Hello everybody. Now we'll see the honey. During the last lecture, we have studied the milk. So now what is honey? What are the possible adulterants which people can add to the honey? How that adulterants can be detected if present in honey? And at the end, we are going to find out the reducible sugar from the honey. The second experiment is there for us in the practical in analytical chemistry. Okay, so as we know that honey is a sugary substance produced from the nectar of flower and stored in the comb by worker honeybee. The flavor of honey depends upon the nectar. Honey is extracted from the comb, strained, and marketed. Raw honey contains an enzyme called as glucose oxidase and which produces hydrogen peroxide, a mild antiseptic when combined with the water. Okay, so honey has a capacity to retain the water and is said to improve the food assimilation and use for the chronic and infective intestinal problems. Honey also has a medicinal value, and the composition of honey changes depending upon the its ripeness. What is composition of honey? Honey contains moisture, glucose, sucrose, fructose, maltose, mineral matter, and protein. Pollen is invariably present in the comb honey, but may be present in some honey which have been very fine strand. Okay, so honey contains larger quantities of fructose, about 38%, then glucose, about 31%, and sucrose constitutes about 2% of the total sugar content. So diastase and invertase are the two important enzymes present in honey. Diastase is responsible to convert starch to dextrin and sugars. Invertase converts sucrose to the sucrose from the nectar to the glucose and fructose. So it is the only food that will not spoil, and as it has a low moisture content and high osmotic pressure, microorganisms such as bacteria cannot survive in this medium. So it is hygroscopic and absorbs the water from atmosphere. Hence, it is stored in the well-sealed container. It contains about 83% of sugar, which is also expressed as 83-degree Brix. The degree Brix unit indicates the gram of sucrose dissolved in 100 gram of solution. So it is a common unit used to describe the sugar content from the honey. Maple syrup, fermenting grape juice. Okay. Now, modes of carbohydrates in the honey are monosaccharide with more fructose than the glucose. Other disaccharide present in very small quantities are the sucrose, maltose, isomaltose, nigerose, turanose, and maltulose. A small quantity of higher sugar, oligosaccharides, and dextrins are also present in honey. It is acidic with the average pH of 3.9 due to the presence of gluconic acid and other acids like citric, formic, lactic, malic, butyric, oxalic, and succinic acids are also present in the honey. Some trace elements, like in our day-to-day life, we find it in the different vegetables also, and we get from different sources. So in the same way, some trace elements like chromium, nickel, zinc, lithium, tin, and lead is also found in the honey. So enzymes found in honey are diastase, invertase, glucose oxidase, and catalase and acid phosphatase. Small quantity of protein and free amino acids are present, among which proline is the dominant. Okay, and vitamins are also there, but that are in very small quantity. So the aroma and flavor that are attributed to the acid, sugar, and other volatile compound in honey, which include the variety of C1 to C5 aldehyde and alcohol esters and different compounds like this. So if we see the honey is actually the nectar collected by the honeybees, they carry that nectar to the honeycomb. Over there the different processes occur, and there is a formation of honeytech press. So whenever the honeybee carries the nectar, so whatever contents are there present in the nectar, that comes in the honey. So hence such a trace elements are also comes in the honey. Okay. So the honey is having different kind of colors depending upon the sources from which nectar is collected, and also the time of collection of nectar also plays very important role, because in the different zone of the countries or on the planet Earth, so climatic conditions are different, flowers are different, plants are different, and accordingly the contents present in the flower, nectar, and the plants are also different. Hence such a variation in the honey color and the taste is also observed. So here the table is given moisture, unripened honey and ripened honey, how the content varies over there. So it is found that the moisture is 19.8, but in ripened is 18.4. So you can go through this table. Okay, it's not much complicated thing. Mature honey and immature honey किंवा आपण म्हणूयात ripened honey and ripened honey, याच्यामध्ये जो फरक आहे हा त्या ठिकाणी आहे, आणि त्याच्यामुळे त्याच्या टेस्ट मध्ये सुद्धा त्या ठिकाणी फरक पडतो, आणि त्याच्या मेडिसिनल प्रॉपर्टीज ज्या आहेत त्याच्यात देखील त्या ठिकाणी काय होतो, फरक ऑब्झर्व केला जातो. So after that we are going to see what are the adulterants or possible adulterant, what is definition of adulterant, and how that adulterants from the honey can be detected. So it is a carbohydrate rich syrup which is a popular sweetener and folk medicine used in the ancient time. So the increasing demand of honey in the market has led to the adulteration of honey and production of synthetic honey to increase the profit margin. So hence, honey adulteration is the act of adding substances like a dextrose, glucose, molasses, sugar syrup, corn syrup, flour starch, and etc. to the honey for the economical benefit of the people who are marketing the honey or selling the honey in the market. Okay, such a malpractice they are doing, and it is subjected to the adulteration with the inexpensive sweeteners like syrups, high fructose corn syrup, then inverted syrup, then high fructose inulin syrup is also there. So such syrups are always added to the honey and to increase the quantity of honey. Okay, so this is what they are doing. So that can be detected with the help of GCMS or FTIR technique. That is possible to detect. Even some more techniques are there by which we can near infrared reflectance spectroscopy is also helpful to detect this thing. Okay, liquid chromatography is also used, and gas chromatography is also used to detect these nuts. Starch-based sugar syrup, the produced from the beet or cane is also used in the adulteration of honey. They can be detected by using isotope ratio mass spectrometer in combination with the elemental analyzer. So water, a cotton bit dipped in the water honey when lightened with the matchstick. If you want to detect the water, okay. So if we dip the cotton in the honey, take out, and if we try to burn it, it easily burns. But if honey contains the water, in that case, it is not producing cracking noise, and it not burns such easily. Okay, indicates the presence of water over there. People also add the sugar or jaggery. So Fahey's test is used for that. Or in this test, we are going to take 5 ml of honey. To this, we are going to add 5 ml of ethyl ether or diethyl ether. Then we will shake it. We will remove that ether layer into the petri dish, evaporate that diethyl ether, and we will add 2 to 3 ml of resacinol. The appearance of cherry red color indicates there may be presence of sugar or jaggery in the glucose or sugar syrup or high fructose corn syrup can also be detected. So we are going to take 1 tablespoon of honey to a glass of water. If honey disperses very instantly into the water, indicates the presence of glucose or sugar syrup. It not, then because what happens, the pure honey is settled down at the bottom, and because that is very thick material. Okay, and in this way we can distinguish the presence of glucose or sugar syrup in the honey. So again, one more test is there. We can take 1 tablespoon of honey, and we will add some water and 2 to 3 drops of vinegar. So if the mixture results in the foaming, it indicates the presence of adulterants in the milks. Okay, so presence of adulterant in the honey. In this way, we can detect the adulterant from the honey. And these are the possible adulterant which people are adding to the honey. Okay, and that is also this test can be taken for the available honey in the market. Okay, we know Dabur honey is there, then Patanjali honey is there, and different kind of honey products are there from which we can do this test. And we will try to, or we can find out the such a substances from the honey. After this lecture, I will proceed to the analysis of reducing sugar in the honey by choleferricyanide method. So that is the sucrose, what we are going to find out. That I will continue in the next lecture. Thank you.